Hello learner, welcome to our lesson 2, cell structure and functions. Let us begin with cell structure and functions as seen under a light microscope. The structures within a cell are called organelles, and they perform specific function within the cell. So what are the cell organelles in plant and animal cell under light microscope? So these, a cell membrane, cytoplasm, and a nucleus can only be seen in an animal cell. For a plant cell, a cell membrane, nucleus, cytoplasm, chloroplast, vacuole, and a cell wall can be seen in the case of a plant cell, that is under light microscope. We move to the electron microscope, which is capable of magnifying up to 500,000 times, and the specimen is mounted or placed in vacuum chamber through which an electron beam is directed, and the image projected on photographic plate. However, it has a disadvantage that it cannot be used to observe living specimen. It is only limited to dead specimen. The advantage is high magnification and resolution power. Resolving power or resolution power is the ability of the microscope to distinguish between or differentiate between separate things which are very close to each other. The high resolving power makes the EM a very important research tool in microbiology. Through the electron microscope, very fine details of the cell can be seen. So what can be seen in the electron microscope? We have talked about greater details of the cell being seen. So we have the cell wall, which was not visible in the light microscope, mitochondrion. We also have the Golgi body, the nucleus. The entire of this is the nucleus. And then we have, or the whole of this is the nucleus. This black is the nucleus. We have the nucleolus surrounding the nucleus, the endoplasmic reticulum, ribosomes, and lysosomes, in addition to those that were seen under the light microscope. That is for a plant cell. In an animal cell, under electron microscope, this can be seen. We have the nucleus and the nucleolus, the ribosomes, we have the vacuole, mitochondrion, the endoplasmic reticulum, secretory vesicle, the centriole, Golgi body, rough endoplasmic, and cytoplasm or lysosomes. So be noting those structures that are present in a plant cell but absent in an animal cell. For example, we can see centrioles and the vesicle in an animal cell, but a plant cell lacks such. When it comes to a plant cell, you can see it has a cell wall and a chloroplast and a very large central vacuole, which is absent in an animal cell. An animal cell has a small vacuole, but the vacuole of a plant is very big. So what are the differences between light and electron microscope? Light uses light to illuminate or to brighten or just say to lighten up the specimen whereas electron microscope uses a beam of electron to illuminate the specimen. In light microscope, glass lenses are used for magnification, but in the case of electron microscope, electromagnetic lenses are used. Light microscope has low resolving and low magnification power, but electron has high resolving and low magnification power. The specimen under view in light microscope can either be dead or living, but electron microscope deals only with dead specimen. In light microscope, normal dyes are used for staining, whereas electron microscope uses complex dyes for staining. The light microscope, specimen mounted on the slide is placed on the stage in the open, but in the electron microscope, the specimen is mounted on a grid and placed inside a vacuum. So what are these organelles found in the cells of plants and animals? First, we have the cell membrane, also known as the plasma membrane or plasma lemma, and contains 3 layers when viewed under the electron microscope. One layer is the phospholipid, which is sandwiched between 2 protein layers. It is flexible and has pores. The cell membrane does the following roles: encloses the contents of the cell and allows for selective movement of materials in and out of the cell, because we've talked about the pores, which allows small substances to pass through, but not large ones. Substances are transported across the membrane by active transport and diffusion. The other part is the organelle cytoplasm, and it consists of fluid medium in which chemical reactions occur, and has organelles and other inclusions like starch, glycogen, fat droplets, and many other dissolved substances. The cytoplasm is not static, but constantly moves, a movement called cytoplasmic streaming. It provides a suitable medium for cellular reactions to take place. We move to the ribosomes. They are spherical in shape. Some are bound to the endoplasmic reticulum. Some are scattered within the cytoplasm. They are manufactured in the nucleolus of the cell and contains protein and ribonucleic acid. The role of ribosome is protein synthesis or manufacture proteins. The endoplasmic reticulum, abbreviated as ER, and consists or is a network of tubular structures extending throughout the cytoplasm of the cell and serves as network of pathways through which materials are transported from one point to another in the cell. And there are 2 types of ER, one with ribosomes, which is called the rough endoplasmic reticulum, and the one lacking proteins called the smooth endoplasmic reticulum. The rough ER transports proteins, while the smooth endoplasmic reticulum transports lipids. So this is the anatomy of the ER. So rough, you can see there are proteins, and the smooth has no proteins. And then surrounding it is the nucleus. We move to the lysosomes, and they are vesicles containing lytic enzymes whose role is breakdown of microorganisms, foreign macromolecules, and damaged or worn out cells and organelles. We move to Golgi bodies, and are thin plate-like sacs arranged in stacks and distributed randomly within the cytoplasm. They are involved in packaging and transportation of glycoproteins. They also produce lysosomes. The nucleus. The nucleus is surrounded by a nuclear membrane, which is a unit membrane. The nuclear membrane has pores to allow materials to move to the cytoplasm. Nucleus contains protein and nucleic acid, DNA or deoxyribonucleic acid, and ribonucleic acid. Chromosomes are found in the nucleus. These chromosomes carries genetic information in form of genes or DNA. The nucleolus is located in the nucleus but is only visible during the non-dividing phase of the cell. So the role of the nucleus actually is to control all the activities of the cell. But it also, the nucleolus is the one manufacturing ribosomes. We move to mitochondrion, and this is sausage shaped to provide sites for respiratory reaction that produce energy for the cell. That is why mitochondria is referred to as powerhouse of the cell. It is because it yields energy. It has two membranes, the inner, which is highly folded into cristae to increase the surface area for respiration. So this is the inner membrane, highly folded into cristae, providing larger surface area for respiration. Then we have the matrix and of course the outer membrane. So the arrangement and number of mitochondria in a cell depends on the cell energy requirement. Cells that require large amounts of energy contain numerous mitochondria. These cells include the muscle cell, so that it can contract and relax to bring about movement, sperm cell, the apical meristem, that is the growing regions in a plant, such as the root tip and the shoot tip, and also the kidney cells in animals. So you can see here it is mitochondrion because we are talking about one. When there are many, they are called mitochondria. Mitochondria are self-replicative, that is, they can divide to form new ones. The other structure is the centrioles. They are rod-shaped, found outside the nuclear membrane, and form spindle fibers during cell division in animals. So they are only found in animals. They also form cilia and flagella in cells and organisms where these structures are found. So cilia and flagella are for movement, and they are found on the surface of the organism. So they are involved in movement. The vacuole is the other part, and plants have large, permanent, and central vacuole that contains the cell sap where sugars and salts are stored. The cell sap contributes to osmotic properties of the cell. animal cells contain small and temporary vacuole, and in unicellular organisms, food vacuoles store and digest food, whereas contractile vacuole excretes excess water, as in the case of amoeba. We move the chloroplast, and they are found in plants or photosynthetic cells. Each chloroplast contains an outer unit membrane enclosing a series of interconnected membranes called the lamella. So, at various points along their strength, the lamella form stacks of discs called the grana, where light stage of photosynthesis takes place. So the lamella are suspended in the stroma. This stroma is the site for dark stage of photosynthesis. Chloroplasts have a green pigment that traps light for photosynthesis. The cell wall. This is the rigid outer cover of a plant cell and lower organisms, composed of cellulose fibers that gives a plant cell a definite or a regular shape. It also provides a mechanical support and protection against mechanical injury. So we want to compare between plant cell and animal cell after having discussed almost all the organelles. So a plant cell has a cell wall and a cell membrane, but an animal cell is lacking a cell wall, so it has a cell membrane only. So in plant cell, the nucleus is at the periphery, but an animal cell has nucleus at the center. Plant cell has chloroplast. Animal cell lacks chloroplast. Plant cell has a large central vacuole which is permanent, but animal cell usually have no vacuoles. In case the vacuoles are present, they are small, temporary, and scattered within the cytoplasm. A plant cell is larger, whereas animal cell is usually small. Plant cell is also irregular in shape, or it has a definite shape as I had shown you, but for an animal cell, the shape is irregular. There are no centrioles in plant cell, but present in animal cell. Plant cell stores food in form of starch, oils, and protein, whereas animal cell mostly stores in form of glycogen and fats. So that marks the end of the lesson. So this is an assignment for you. Nice time.